Chapter 17. Immune activation in the gut. Immune activation in the gut reflects the condition of the material presented to the intestinal barrier rather than an isolated malfunction of the immune system itself. The epithelial surface is continuously exposed to both dietary substrates and microbial byproducts, and the form in which these arrive determines the level of immune signaling that is generated. When digestion proceeds in sequence, most nutrients are reduced to defined molecular units and absorbed before reaching the distal intestine. The barrier encounters a controlled set of inputs, and immune activity remains within a stable range. When digestion is incomplete, larger fragments and increased microbial metabolites reach the epithelial surface together, amplifying signaling within the system. The structure of the substrate is the first determinant of immune interaction. Small peptides, amino acids, and simple sugars interact with the epithelial surface in predictable manner and are rapidly transported across the membrane. Larger fragments persist in the lumen and increase contact with the barrier. This prolonged interaction changes how the epithelial layer responds, increasing signaling intensity and altering the local environment. The difference lies in the form of the substrate rather than its identity. Microbial activity compounds this effect by generating additional compounds from the substrates that reach it. When fermentation increases due to incomplete digestion, microbial byproducts accumulate and interact with the epithelial surface alongside residual dietary fragments. This combination changes the chemical conditions within the lumen and increases the complexity of signals encountered by the barrier. The immune system responds to this combined environment, integrating signals from both microbial metabolism and unprocessed substrates. The location of these interactions influences their impact. When digestion is aligned, most substrate exposure occurs in distal regions where the system is adapted to handle microbial activity. When upstream processing fails, substrates appear earlier in the intestine, shifting immune interaction proximally. This increases exposure in regions where the barrier is not adapted to sustain contact with large amounts of microbial byproducts and partially digested material, amplifying the response. Motility and luminal pressure also contribute to immune activation by altering the duration and intensity of exposure. Increased gas production and irregular transit patterns change how long substrates remain in contact with the epithelial surface. Prolonged retention increases the opportunity for interaction, while rapid transit can deliver substrates in forms that have not been fully processed. Both conditions reflect upstream variability and influence the level of immune signaling generated. Immune activation, therefore, represents an output of the digestive sequence, shaped by the structure, quantity, and distribution of substrates within the intestine. It arises when the barrier is exposed to material that has not been reduced and absorbed in sequence, combined with increased microbial byproducts generated from that material. The response reflects the environment created by digestion and microbial activity rather than an independent failure of the immune system. Exposure to incomplete food fragments. Exposure to incomplete food fragments occurs when upstream digestion fails to reduce macronutrients into the molecular forms required for membrane transport. Proteins that should be converted into small peptides and amino acids, carbohydrates that should be reduced to simple sugars, and lipids that should be dispersed and cleaved into transportable units remain partially intact. These larger fragments persist within the lumen and increase contact with the intestinal barrier. The epithelial surface is then presented with material that extends beyond its normal handling range, changing the nature and intensity of local signaling. The duration of exposure is determined by motility and the physical state of luminal contents. When digestion is aligned, substrates are reduced and absorbed quickly, limiting the time they remain in contact with the epithelial surface. When reduction is incomplete, these fragments remain within the lumen for longer periods, increasing the opportunity for interaction. Irregular motility further alters this exposure by either prolonging retention or delivering partially processed material more rapidly into downstream regions. In both cases, the barrier encounters substrates that have not been prepared for immediate uptake. The structural properties of these fragments influence how they interact with the epithelial layer. Larger molecules present more complex surfaces and persist at the interface between the lumen and the membrane, altering how transport systems and surface receptors engage with them. This changes the pattern of signaling within the epithelial cells, increasing activity in pathways that respond to luminal content. The response is driven by the form and persistence of the substrate rather than by its origin. Microbial metabolism amplifies this exposure by acting on the same incomplete fragments. As microbes process these substrates, they generate additional compounds that accumulate alongside the original material. This creates a combined environment where dietary fragments and microbial byproducts interact with the epithelial surface simultaneously. The barrier responds to the totality of this environment, integrating signals from both sources. The location of exposure shifts when upstream digestion is impaired. Instead of being confined to distal regions, incomplete fragments appear earlier in the intestine, increasing contact with sections that are not adapted to sustained exposure. This alters the distribution of interaction along the digestive tract and changes how different regions respond to luminal content. Exposure to incomplete food fragments, therefore, reflects a failure of the digestive sequence to produce absorbable forms. It increases both the duration and complexity of interaction at the epithelial surface, altering signaling within the intestinal barrier. The extent of this exposure is determined by how much material escapes upstream processing, how long it remains in the lumen, and where it accumulates within the digestive tract. Barrier stress and immune signaling. Barrier stress develops when the epithelial surface is exposed to substrates and byproducts that exceed its normal processing range, and immune signaling increases as a direct response to that exposure. The intestinal barrier is designed to interface with defined molecular forms that can be transported across the membrane or cleared efficiently. When digestion proceeds in sequence, most substrates are reduced to these forms before reaching the epithelial surface, and contact remains controlled. When upstream processing fails, larger fragments and increased microbial metabolites persist in the lumen, extending both the duration and intensity of contact with the barrier. The physical state of luminal contents determines how this stress develops. Poorly reduced substrates remain in larger, more complex forms that interact with the epithelial surface over a wider area and for longer periods. These fragments alter the local environment at the membrane interface, changing how epithelial cells respond to what they encounter. Instead of rapid transport and clearance, the system shifts toward increased signaling, reflecting the presence of material that cannot be processed through standard pathways. Microbial metabolism amplifies barrier stress by generating additional compounds from the same substrates. As fermentation increases, these byproducts accumulate alongside partially digested material, creating a combined environment that differs from the conditions present during coordinated digestion. The epithelial surface responds to both sources simultaneously, and the signaling intensity reflects the total burden of luminal content rather than a single input. Motility and pressure further influence this interaction. Increased gas production and irregular transit patterns change how long substrates remain in contact with the epithelial surface and how they are distributed along the intestine. Prolonged retention increases exposure time, while uneven distribution creates localized regions of higher stress. These mechanical and temporal factors contribute to variability in how the barrier experiences luminal content. The location of barrier stress shifts when digestion is impaired. Instead of being concentrated in distal regions, exposure to complex substrates and microbial byproducts occurs earlier in the intestine. This increases interaction in regions that are not adapted to sustained exposure, amplifying the signaling response and altering how the barrier functions in those areas. Barrier stress and immune signaling, therefore, arise from the interaction between incomplete digestion and microbial metabolism. They reflect the condition of the luminal environment rather than an isolated immune process. When digestion is aligned, exposure remains limited and signaling stays within a stable range. When it is not, increased and prolonged contact with complex substrates and byproducts drives a higher level of immune activity as the system responds to the environment it encounters.